Welcome back to Concepts of Biology. We'll read Chapter 3, Cell Structure and Function, Microscopy, Electron Microscopes. In contrast to light microscopes, electron microscopes use a beam of electrons instead of a beam of light. Not only does this allow for higher magnification and thus more detail, like in Figure 3.3, it also provides higher resolving power. Preparation of a specimen for viewing under an electron microscope will kill it. Therefore, live cells cannot be viewed using this type of microscopy. In addition, the electron beam moves best in a vacuum, making it impossible to view living materials. In a scanning electron microscope, a beam of electrons moves back and forth across a cell's surface, rendering the details of the cell surface characteristics by reflection. Cells and other structures are usually coated with a metal like gold. In a transmission electron microscope, the electron beam is transmitted through the cell and provides details of a cell's internal structures. As you might imagine, electron microscopes are significantly more bulky and expensive than our light microscopes. Figure 3.3. Salmonella bacteria are viewed with a light microscope. This scanning electron micrograph shows Salmonella bacteria in red invading human cells. Cytotechnologist. Have you ever heard of a medical test called a pap smear? Figure 3.4. In this test, a doctor takes a small sample of cells from the uterine cervix of a patient and sends it to a medical lab where a cytotechnologist stains the cells and examines them for any changes that could indicate cervical cancer or a microbial infection. Cytotechnologists, cyto meaning cell, are professionals who study cells through microscopic examinations and other laboratory tests. They are trained to determine which cellular changes are within normal limits or are abnormal. Their focus is not limited to cervical cells. They study cellular specimens that come from all organs. When they notice abnormalities, they consult a pathologist who is a medical doctor who can make a clinical diagnosis. Cytotechnologists play vital roles in saving people's lives. When abnormalities are discovered early, a patient's treatment can begin sooner, which usually increases the chances of successful treatment. Figure 3.4. These uterine cervix cells, viewed through a light microscope, were obtained from a pap smear. Normal cells are on the left. The cells on the right are infected with human papillomavirus. Cell theory. The microscopes we use today are far more complex than those used in the 1600s by Anthony van Leeuwenhoek, a Dutch shopkeeper who had great skill in crafting lenses. Despite the limitations of his now ancient lenses, van Leeuwenhoek observed the movements of protists, a type of single-celled organism, and sperm, which he collectively termed animalcules. In a 1665 publication called Micrographia, experimental scientist Robert Hooke coined the term cell, from the Latin cella meaning small room, for the box-like structures he observed when viewing cork tissue through a lens. In the 1670s, van Leeuwenhoek discovered bacteria and protozoa. Later advances in lenses and microscope construction enabled other scientists to see different components inside cells. By the late 1830s, botanist Matthias Schleiden and zoologist Theodor Schwann were studying tissues and proposed the unified cell theory, which states that all living things are composed of one or more cells, that the cell is the basic unit of life, and that all new cells arise from existing cells. These principles still stand today. This ends Chapter 3, Section 3.1. In our next video, we'll read Chapter 3.2, Comparing Prokaryotic and Eukaryotic Cells.